Sophia Mofopoulou,
Yes.
who is currently at University College London and a student of Vincent Blagnol, who I think he will be joining us possibly at some point.
Maybe in the following weeks, I don't know.
In the following weeks, possibly. Your field is statistics. You've
Yes.
done
A bit of bioinformatics.
You've done statistics and bioinformatics. Now you're doing a PhD with Vincent, and this is the topic of your, or part of your PhD, I guess.
Yes. So I'm a final year PhD at the UCL Genetics Institute, and today I'm going to talk to you about my work about metagenomic diagnostic testing using a Bayesian mixture model. Okay, so my main interest is about the human metagenomics in the context of human health. So, for example, we know that viruses have been known historically to cause diseases, but studying the human microbiome, we have found out that they can also chronically infect healthy individuals as well. Or we are aware that both bacteria and viruses are implicated in the pathogenesis of inflammatory disease. And also another interesting area of research is the interaction between a viral infection and a gene mutation. So understanding the human microbiome is very relevant to human health. There are various of research themes in human metagenomics, and one may simply want to perform an exploratory analysis in a sample, or to estimate the diversity within a community, or compare metagenomes across samples, or at different time points. My focus in my PhD is to actually be able to accurately identify all the organisms that are present in a sample, as well as infer the relative abundances. And I want to do this in a connection to clinical diagnostics. That means to be able to test for presence or absence of a pathogen. Okay, so clinical diagnostics, what we mean with this term is just the accurate detection of pathogens. And this is where deep sequencing is of great help, because it has the capacity of detecting... Oh, sorry, should I... Okay, is it better? Yeah. So deep sequencing has the ability of detecting pathogens at concentrations lower than the ones that are detectable by PCR, and also due to the unbiased nature, we can detect pathogens that we didn't know beforehand that they're associated with a specific disease, so we might not be testing for them, but they might be revealed with deep sequencing. And also another relevant point is that still in infectious diseases, there are a lot of there is a great percentage of the diseases that have unknown causes. So, for example, in gastroenteritis, half the cases are of an unknown etiology. It's even higher in encephalitis, and so on. Okay, so I would like you to imagine an example of a patient who is infected and is suffering from infection or a disease who is caused by infection, but the traditional tests are not able to locate the pathogen, to detect the pathogen. And as an example, as a real clinical application, last summer, Judith Brewer, who is a professor of virology in UCL, contacted Vincent and I about an 18-month immunosuppressed boy that had encephalitis. However, the traditional techniques weren't able to find the pathogen, and I mean they performed PCR for bacteria and viruses. However, the histological examination of the brain showed that there was an infection. So here I have just a couple of figures from the paper we're submitting next month. And the first is the MRI taken 5 days after the onset of the encephalitis and a month after the onset, and you can see the swelling of the brain area. And also the other picture shows the staining that is showing the infection. I'm going to go through the method, and then in the end I'm going to tell you what we found out. Okay, so typically when somebody wants to perform a reference-based taxonomic assignment, what they do is that they have the clinical sample, we sequence it, and then we try to allocate the reads to species. And after the initial annotation of the reads and the contigs, we are left with thousands of potential species showing to be present. So, as a similar example as the one that Daniel had the first day in your presentation that you did for the mock community, that was a sample that had 20 species, but there was 700 found as present. I did the same thing about one of the famous datasets. So this is a dataset that has, it's an artificial dataset, but it comes from real sequencing reads, and the reads come from 113 species. So after my initial blast, there are over 2,000 species found as present, which is horrible. It's quite terrifying. And this is because of the fact that there is an extensive level of similarity between the various species. And even though in human tissue we do expect to see a diversity of virus and bacteria, we don't expect them to be in the hundreds or thousands. So what we want to do is to be able to have a parsimonious explanation for this complexity. What is the limited set of species that explains this complexity? So yes, so after performing a blast type analysis, somebody has a matrix with homology information where we have approximately 1,000,000 rows where each row corresponds to a read and a couple of thousand columns where each column corresponds to a species. And we have similarity information between each read and each species. This is a largely sparse matrix. And as I said, we want to eventually find the smallest set of species that explains the data. So what people do when they perform reference-based taxonomic assignment is they try simple, straightforward approaches, counting reads or unique counts, or maybe doing something more sophisticated as using weighted counts. There are a couple of statistical methods that try to solve the problem, but the focus is actually abundance estimation. I'm going to talk about two of the methods very briefly. So one is GRAMI, which stands for Genome Relative Abundance Estimation using a mixture model. And so a mixture model is definitely a step towards the right direction. This is what I'm using as well. But using it on its own, in order to be able to decide what species are present, it's not enough. So what the authors do is that they just perform a huge expectation maximization step where all the potential species are the categories of the mixture model, and then they go after the algorithm has converged and they see how many species have non-zero abundances. So I did the same with the famous data set, and the species that had non-zero abundances were a bit less than 400. So even though it's a huge improvement compared to the initial point, it's still far away from the truth. Another method is GASIC, which stands for Genome Abundance Estimation using a similarity information. And the authors think that the main problem is indeed the similarity between the species. So they want to correct the observed abundances, which are the read counts, using a similarity matrix for the references. So in order to achieve this, they have to perform an extra 2 bioinformatics steps. So typically we have to go through some bioinformatic analysis in order to get to the point where we can analyze the data and see what species are there. But they have to simulate reads from each genome in the database and then align against all other genomes. And that means that they have to perform k-squared alignment. So if you have a lot of species in your database, this is a great amount of effort to go through. Okay, so none of the previous methods can actually provide an answer to a clinician of the type there is 99% probability that there is at least one enterovirus in the sample. And this is what we want to be able to do. So we want to be able to explore the posterior distribution of the species given the sequencing reads, and we want to be able to estimate the probability of a species being present in the sample. And having the posterior distribution means that we can actually answer more fancy questions such as what is the probability of having the two specific species being present jointly in the mixture. And we're going to do this using Bayesian model averaging. And even though it's crucial to use both bioinformatics and statistics to accurately analyze metagenomic data, my focus is on the statistics. Okay, so my primary interest is actually in detecting viruses in human tissue. And I'm saying this in order to explain the motivation for using RNA sequencing data. This is because we want to be able to detect retroviruses as well, but we are not in any danger of missing out to other organisms because we're going to get through them through the RNA they make. We use unbiased shotgun sequencing, and so most of the concepts I'm going to describe later on can be fully applicable to DNA mixtures, and it's just the bioinformatics bit that's going to have to be adapted a little bit. So a typical sample is a human tissue. So far I have worked with post-mortem pancreatic samples, a fresh biopsy brain sample, and esophageal tumor samples. The technology we're using is Illumina RNA-seq, and that means that we have as raw data millions of transcriptome search reads, and the repair dent and the length is 100 base pairs, usually. So I have created a bioinformatics pipeline in order to get to the step that I can analyze the data and see what species are present. So I'm going to guide you very quickly through that. So at first I try to remove the PCR duplicates. I keep only the good quality data. If I have a situation where within a pair the reads are overlapping, I merge them to get a single longer end single and longer read, I do my best to filter out the human sequences, and then I perform a high quality, a high confidence assembly. So that means that I don't try to make contigs for both the low abundance and the high abundance organisms. I just try to assemble the high abundance organisms using a high K-mer value. And then I'm going to use both the contigs and the non-assembled short reads to annotate them, and I use BLASTX, and I compare with the protein database. And the figure I have is just to show you that even though the starting point is the raw data are between 20 million and 100 million, by the time I am left with the non-human sequences, I'm between 100,000 and 1 million reads. Okay, so it might seem obvious now that assembly is not always effective, but when I started my PhD 3 years ago, it wasn't obvious to me, so I was just working with assembly output. However, in one of the samples I analyzed, I managed to detect a very weak signal of a murine leukemia virus. And when I say weak signal, I mean two very short contigs. So I have here an illustration of the genome, and purple lines are the genes of the virus, and the short red lines are the contigs I managed to assemble. But when I went back and I looked at the rest of the data, I saw that there were around 50 reads that were matching this virus. So the data were supporting the presence of this virus a lot more if I took into account both the contigs and the unassembled reads. This virus was not something biologically interested, it was just a contamination, but it just opened my eyes that I have to use both assembly and the remaining reads. So this is a difficult problem to solve because we have a database complexity, so we have a lot of species in the database and they can be highly similar, but also they're incomplete, so there are a lot of species that are not there yet. And the rare signals, so I haven't explained this yet, but with viruses it's very typical that we might get the samples quite some time after the phase of the acute infection. So by the time we get the data, there can be maybe only traces of the virus left. So I'm interested in being able to detect very low abundant organisms in the sample. Okay, so a mixture model. So a mixture model fits perfectly with metagenomic data because it's when we have a situation where we have a population that consists of several subpopulations, and each observation comes from one of these subcategories, but we have lost the membership information throughout the observational process, we can model it by using a mixture model. So it is defined by the mixture weight and the component distribution. And the mixture weight is the multinomial probability that the read comes from a species. And then we have the generative probability. So given that the read comes from a species, what is the probability that's generated by it? And I incorporate into this estimation of the generative probabilities both the length of the genome, or the gene if I'm looking at RNA-seq, and the number of mismatches between the read and the protein. I also have added in the model one category for the collective of unknown species in order to be able to deal with the reads, the sequences in my sample that come from unknown species. So the probability for observing read die is just simply mixture weight times the generated probability summed across all species. And for that we can get the dataset likelihood, which is the same sum but also multiplied over all reads. Okay, so what we want to be able to do is to compare several sets of species and see what best explains the data. So the way to do this, what we use is marginal likelihood, which is central in Bayesian inference and model comparison. And in order to estimate the marginal likelihood, we have to integrate out the mixture weights. And the basic Monte Carlo way to do this is just to simulate a lot of weights from the prior. So I didn't mention this, but the prior here is a conjugate Dirichlet, and average the likelihood. However, simulating from the prior is very inefficient from a computational point of view because a lot of the samples we're going to get are going to be outside the regions of high likelihood. So the approach I'm using is just important sampling. I'm not saying that this is the best way. I mean, it's a very difficult problem to do, estimate the marginal likelihood, but this is the way we're doing it right now. So I use a Gibbs sampler step to approximate the posterior distribution with a multivariate normal, and then I generate samples from this approximating distribution. And then I weight these samples to compensate for the use that for the fact that I'm using the approximating distribution. Okay, so what I have described so far is in the situation where we know how many species are in a sample. So this is the assumption was that we know exactly how many categories we have in the sample. But the real difficult problem is to be able to decide how many species we have. So in order to do that, we have to consider all the potential subsets, all the possible combinations of species. And in reality, this problem is even more difficult because only a small subset of the species is going to be actually present in the mix. So this is a very large parameter space that we have to explore. So regarding the estimation of the marginal likelihood, I also incorporate a likelihood penalty term in order to limit the number of species and enforce the parsimony of the model. So this is a tuning parameter, and because I'm interested in hunting a rare signal, I have tuned it to be very sensitive. So my penalty term corresponds to 3 short reads, to matches from 3 short reads. Okay, so what is the best way to explore this huge set of species? It's a Monte Carlo Markov chain, and here I have a table to show what happens at each iteration. So at one iteration, we might consider as present species 1 and species 2, and then we might remove species 2 and keep species 1, remove species 1, keep species 2, and so on. Okay, so let me summarize what I do. I consider at each iteration that I have a set of species, and because the k is known, I'm able to both estimate the abundances and estimate the marginal likelihood. And then I use a Monte Carlo in order to, a Markov chain, to be able to walk across the space. So how does it perform? Well, very poorly. I mean, the mixing is terrible. You can see that the chain gets stuck for long periods of time. So what I have plotted here is a trace plot of the log likelihood across the second half of iterations. And you can see that it gets stuck for very long periods of time. And this would take ages to converge. So the reason this is happening is because while it might seem to you that adding or removing a species, it's a simple change, the addition or the removal of each space might have a huge impact. So we might have to perform more subtle changes, such as swapping two species at the same time, or swapping two groups of species. And you want to be able to use multiple machines in order to speed things up. So we need a computation scheme that will both have the flexibility to explore the parameter space and use parallel computing. So for that reason, we perform a parallel MCMC. So we run multiple chains in parallel, and I do this in the context of parallel tempering. So I use tempered sequence of distributions, and neighbors will communicate, neighboring chains will communicate at various points with each other and try to exchange information. And the main idea is that the chains that you have at higher temperatures will be more able to explore the space easily because the likelihood is flatter. And I have two schematics here to show how it works. So the first one shows 5 chains at different temperatures. And for some time, the chains will mutate, and then they will stop and talk to each other, and maybe they will swap values as well. And then I have plotted 4 chains from my parallel tempering. And you can see what I want to show is just that that all the chains, hot and cold, they will eventually converge. And in order to compare with how the single chain does, I have created the same plot, the trace plot of the log likelihood from a parallel tempering for the target distribution. And you can see that it's mixing, the mixing is much better. Okay, so that was the description of the method, and now it's time to check how it performs with data sets. And I've used as benchmark data set the famous artificial one. So this is the data set that I was just describing before that has 113 species. So we know exactly how many species are there, we know how much of them, and we know that they have been, these data sets have been designed to model real metagenomic communities. So we have several strains of the same species or several species of the same genus in these data sets. And I applied my method to the highest complexity data set, which is the one that has no dominant species. All abundances are roughly in the same range. So this is a data set that doesn't really share the same characteristic as the human samples that I'm looking at, which are mostly sterile. So I wouldn't expect to have 100 species in brain, for example. Okay, so the results. So the method managed to detect successfully all 113 species. and 110 of them were the exact strain level, 3 were different strain but the same species. And in total, at a posterior probability of 0.9, I detected 117 species. So to compare with the other methods, we started with 2,000 with all potential species. The simple mixture model finds 400 species, and this can find that with 90% support 117 species. So the extra 4 species I found were actually different strains of the same species that were actually there. And I think that my method detects these extra species because of the likelihood term penalty. So on how sensitive I designed the method to be. So I still tune it to be sensitive. So I would accept a species if I would, not always, but most frequently accept a species if it was supported by 10 reads. And I could also estimate the relative abundances, so this is of secondary importance to me, but the method still does it quite well. So I have the true abundances, and I have the estimates of the abundances, so I can calculate the relative root mean square error, which is sensitive to outliers, and the average relative error. And it seems to be doing okay. And then I compared it with the 2 other methods I mentioned previously. So the method is doing a bit better than GRAMI, which is the mixture model, and a bit worse than GASIC. But however, the GASIC is trying to solve a different problem. So they assume that they know the exact composition of the community, and they just work with this exact 113 species. On the other hand, my database had 2,700 species. But in the specific context, GASIC estimates the abundances better. Okay, and now it's the time for the ugly truth. There are, of course, as always, practical considerations. So the marginal likelihood estimation was taking time, so I've also tried to let go of the Bayesian approach and maybe use the maximum likelihood estimator of the marginal likelihood. And indeed, it saves a lot of computational time, and it seems to work okay in practice. I'm limited in the number of iterations I can run in a reasonable amount of time. So I do an approximating step at the beginning, which is a GRAMI type, so I perform a QGM in order to lower the species to the low hundreds, and then keep this in my MCMC loop. I'm not sure yet how to account for insertions and deletions, so my mismatches model doesn't take into account the fact that we can have indels. The number of chains that I use is quite small, it's 16 chains, but the more chains I add, the greater is the communication burden, so the algorithm becomes more slow. And I have implemented this in R using the RMPI package, which offers an interface with OpenMPI. And for the end, I kept the results from the clinical sample I mentioned before about the boy that had encephalitis. And so 2 months after the onset of the encephalitis, there was a brain biopsy was performed. And the data consists of 20 million per reads. And after the bioinformatics processing, I was left with 75,000 reads. So the Bayesian mixture model took 6.5 hours to run, and it identified 2 viruses. So one was not interesting at all because it was the positive control for Illumina sequencing. And the other one was an astrovirus. So I found 49 reads assigned to an astrovirus, and with posterior probability 1. And then the clinicians managed to reconstruct the full genome of the astrovirus. So it was a novel strain, different to the one that was in the database and to the one I detected. And here I have a similar schematic just to show that where the genes of the astrovirus are and where the reads, which are the blue short lines, were coming from. Okay, and just to sum it up, there is a lack of statistical methods that can test for presence of pathogens in a sample, or even to be able to create an accurate catalog of what species are present in a sample. And my method is limited by the product of reads and species. However, the Bayesian framework is very useful to be able to explore the large parameter space and deal with the fact that we have a model uncertainty. And of course, deep sequencing is an exciting way to detect pathogens that elude conventional testing. Thank you. I would like to thank my supervisors as well. Yeah.
Thank you. Yes.
So this astrovirus that you found, would the PCR primers traditionally used for diagnosis have picked up that virus?
Sorry, what?
This virus that you found in the end, would the traditional PCR primers would have picked that up?
No. So, I mean, at the beginning there was performed a PCR for the astrovirus as well. There was a PCR for the astrovirus as well, but for a different strain.
Okay.
So for the human astrovirus, while the one that was detected, I think it was more similar to a bat astrovirus. So the primers that were used were not similar to.
That's a primer question, because my model, and I'm asking people like Nick to correct me, is that you need a lot less material and a lot less abundance to be picked up in a PCR-based assay. That's why the whole sort of 16S amplicon works with much lower input amounts and actually is far more sensitive than shotgun.
So afterwards, for verification, they managed to pick it up, but I'm just saying that at the beginning they didn't. Yeah, but I mean it's true. They managed to find high levels of the astrovirus in the sample.
Okay.
Yeah, I mean, the point about this is that it's cool because you can diagnose things you're not looking for, things that are very
Of course, I
divergent.
mean, yeah. Nick, I'm not talking against the use of metagenomics.
Really? Yeah, so I just had I thought that was a very nice talk and sort of addressed what Daniel's mentioned earlier on in the week, right? But the actual method that you had for sampling, I think could be made more straightforward. If you, I may not be right, but if you put a Dirichlet prior on your mixing proportions, then you can marginalize them out, and you can do actually a fairly simple Gibbs sampling strategy, and you wouldn't need all the.
But I do a Gibbs sampling in order to estimate the abundance. But how can I get the marginal likelihood?
You actually you marginalize out the abundances. So you just consider the assignments of reads to the different species. You don't have an explicit mixture proportion, and then it's actually very similar to the sampling algorithms people use in Dirichlet processes and such.
Okay.
And it might actually make your life a whole lot easier.
Okay. I was not aware of doing that. Yeah.
So you get you only get the beginning and the end of the virus.
Yes.
Do you know why that is?
No, not really. I mean, that was odd, but.
Is it just they're more homologous to
or maybe it was the one that was mostly expressed because it was at the protein level. So maybe these proteins were more expressed in the RNA-seq data I had.
I mean, do you now have the true sequence of the?
I don't have it, but it exists. Yeah, I mean they have reconstructed the whole genome.
So maybe you can repeat the experiment and see what you get.
So maybe I can look at the rest of the reads and see whether they map now to the definitely. Actually, I could even look at because I have this unknown bin. So maybe I can look from the reads that have been assigned to the unknown bin how many reads maybe map to the novel astrovirus, but I haven't done that yet.
Maybe I can ask a question. I didn't fully understand all the sampling going on, but there was some important sampling
Yes.
in the middle of that.
So what I do is I perform for a specific set of species. I perform a Gibbs sampler. So I have a number of samples, and then I use this to approximate the posterior by a multivariate normal. And then I sample from this distribution, and then I perform. So this is my important sampling distribution, the multivariate normal.
Okay, so the main. Is it the case then that the samples you generate for inference are basically from this important sampling step?
Yes.
Yeah, and the rest of it is kind of prior,
Yes.
some preliminary work in order to get yourself a decent proposal. Is that the
Yes.
case? Okay. I mean, so how did you find that? I mean, this important sampling is, wonderful if it works, but you end up mostly throwing everything away.
So I think actually I have the opposite problem, that the important sampling distribution that I'm using is very similar to the posterior. So I might end up in a situation of the harmonic mean estimator. So I think
You might end up with
calculating the harmonic mean estimator, because my approximating distribution is so similar to the posterior.
So what you can do in that situation is you can sort of put a bit of extra variance on your
To make it less-
variance matrix,
to
so
make
the
it less-
tails are thicker.
Yes.
I think if
okay.
you don't do that, then you can get some very dangerous
Unstable, yeah.
But that's all you need to do. That's the standard
So,
thing.
so I just need to add some variance to the weights that
Just
I'm.
add a bit more variance.
Okay.
You know, just that's all you
I was
need.
thinking about that. I have also tried to estimate the marginal likelihood using the CHIB estimator, which seemed to work in a similar way, but anyway, I did this a couple of years ago when I started setting my model up, so I didn't go back to the cheap estimator, I just went on with the importance sampling. Okay, but
We're going to
okay.
talk about it.
Yeah.
Is there any further questions? So I think we'd like to say thanks to Sophia once again.
Thank you.